So this is what we're doing in what I'm calling the first round of the race to robust mouse rejuvenation. This experiment, we're calling it RMR1. So first of all, I've told you already, we're doing it starting at 19 months of age. And secondly, as you would expect, given that it's being done by LEV Foundation, we are doing damage repair, we're doing rejuvenation, we're not doing slowing down of damage creation, or at least not much. I'll come to that in a moment. But the big thing, of course, is that very much as I have been saying for the past 23 years, the nature of damage is that it's very multifarious. There are lots of different types of damage that accumulate during life, and that's true whatever your preferred definition of damage, something that we discussed yesterday. The thing is, therefore, you need different methods to repair that damage. And so the key thing that we're doing is we're doing combination therapies. In this first experiment, we are looking at 4 different interventions, and we decided that in the first experiment we would try to be as conservative as possible in how we would choose which interventions to include. In other words, we would try to do things for which the evidence that they would help was as direct as possible. And the most direct evidence in this case is, of course, when someone's taken the intervention individually and already shown in mice, in normal wild-type mice, the same ones that we're using, starting late, that there is some degree of extension of mean and maximum lifespan, and of course health span as well. And the treatments that we have here pretty much hit that mark, they tick that box. So let me just go through them a little bit. Rapamycin is a chemical that how about anyone in the room who hasn't heard of rapamycin? Good, good. All right, so so rapamycin has been shown over the past, I guess 12 years now, 14 years, to, by many, many groups, to quite robustly extend mouse lifespan. And that's true even when you start late. Many of you will know the story that when the intervention testing program tested rapamycin, they had a problem with the formulation of the chow, which delayed the experiment by a year, but for whatever bureaucratic reasons, they'd already got the mice that they were going to use, so the mice were actually 20 months old when they started getting fed rapamycin, and the whole thing worked anyway, much to everybody's stupefaction. And so that started the rapamycin mania that has consumed so many people since. So we're using that. Now, a few groups recently have been doing the next one. Of course, all of you know about the parabiosis mania that started in 2005 when Irina Comboy and her husband Mike and Amy Wagers in the labs of Tom Randau and Irv Weissman at Stanford showed quite robust rejuvenation of old rodents when they were surgically attached to young rodents. And that was a very exciting experiment, but the overwhelming majority of the work that has gone on since then to understand and elucidate and exploit this phenomenon has focused on plasma. In other words, has not focused on the cellular component of the blood. And a few people have decided that that's a bit of a shame. So one thing that was done to change that, to explore the cellular component, was very simple, a heterochronic bone marrow transplant. Of course, you can do bone marrow transplants in mice, same as you can in humans. You use mobilization agent like GCSF, and if you inject a nice lot of cells from young mice, you can get some of the cells that go back into the bone marrow that re-engraft to be the young ones, and the result is that the blood is partially rejuvenated in terms of its cellular composition. So that's one thing that we're doing, because as with rapamycin, this has been shown to work in terms of lifespan by a couple of groups. Another thing that's been done, again by a couple of groups, is to give a virus to mice at middle age that encodes telomerase, the catalytic subunit of telomerase. And rather paradoxically, this also quite robustly extends mean and maximum lifespan. I say it's paradoxical because, as many of you will know, unlike humans, mice express telomerase, mTERT, very promiscuously. Not in absolutely every cell type, but really rather widely. So it was a little bit of a surprise that, the first group that did this was that of Maria Blasco about a decade ago, when they found that, yeah, this actually is beneficial. But it seems, as again, I think Maria gets most of the credit for this, it seems that the natural telomere maintenance machinery of mice is so utterly crap that their telomeres shorten insanely fast. And there's a lot of variation in how rapidly they shorten, and as you all know, the shortest telomere in a cell is the one that makes the cell unhappy. So one way or another, putting in a boatload of extra telomerase seems to be good for them. So that's another thing we're doing. Now, the last one we have slightly less robust evidence for. There is not really, I would say, robust lifespan evidence for this one, but it's not far off that, in terms of the quality and the robustness of the evidence. Navitoclax is a senolytic. It is, of course, and then senolytics are another area of geroscience that has seen enormous attention over the past decade or more. And many of the compounds that have been used have been claimed to have really widespread beneficial effects. There is a certain amount of concern in the community with regard to the reproducibility of some of the work that's been published on senolytics. But our feeling is that navitoclax is a compound that has pretty robust evidence in terms of its benefits. It is toxic to platelets, as was found not long ago. And that means that it's a great candidate for an enormously clever trick that was first thought of by Manuel Serrano, who now works at Altos Labs, but was also in Spain a long time ago, and indeed used to be married to Maria Blasco. The idea is to exploit the fact that senescent cells, by and large, express very high levels of a particular enzyme, beta-galactosidase, that breaks down galactose. And so the initial idea was to encapsulate your drug of interest in galactose, so that even if it went everywhere, it would only actually be bad for the cell it was in if the cell it was in it was expressing a lot of this enzyme. Of course, it's not 100% selective, but it's definitely improves the selectivity, and therefore it's going to make a better senolytic. Sometime after that, Manuel decided that perhaps a better way to do this would be to make an actual bona fide prodrug. In other words, not just to encapsulate the drug in galactose, but to conjugate one molecule of galactose in an appropriate location on the molecule that would inactivate it. And sure enough, that works too. And so this is what we're using in our experiment. We are injecting galactose conjugated navitoclax. So as I've already said, when the treatment starts, namely about 19 months of age, and our goal is to extend mean and max by another year. Now, you may have noticed that we may or may not succeed in that, and towards the end of my remarks, I'm going to be discussing what we're going to do next. But that's the start. And of course, we're measuring lots of health span things. So I've talked about all of the things on the left already. I'm sorry for the slightly illegible font. You may ask, how are we going to know things like whether we're doing the experiment right, in particular in relation to all these interventions, and how are we going to identify antagonistic interactions between them, things like that? And the answer is, by a fucking shitload of mice. We are using 1,000 mice in this experiment, and we're splitting them into 10 treatment groups. So that means 50 mice of each sex for each group. We've got obviously a control group that doesn't get anything. We've got a bunch of groups that get exactly one of the interventions. So in order just to validate that we are doing the thing right. and we've got one group that gets all 4 of the interventions, and we've got 4 groups, each of which get all but one, which is really how we're going to find out about antagonistic interactions. Just want to say a little bit about the controls. The interventions that we're doing are, of course, quite invasive. This is one thing that we felt was absolutely necessary, that in order to do rejuvenation comprehensively, you're going to have to do things like cell therapies and gene therapies one way or another, and thus we are going to have to inject stuff. And injection is, I don't like it much. So we want to make sure that we have some measure of the extent to which the method of delivery is impacting lifespan, or negatively for that matter. And so in each of the groups we have 2 types of control. One is basically the same intervention delivered in the same way, just inactivated in one or another way, and the other one is just not the intervention. So that's something that we think is important. This is an innovation that I'm quite pleased with myself for, because I haven't seen anyone do it before, but it seems to me to be a genuinely useful refinement of the way that lifespan studies are typically done. Of course, the idea here is we do want to know about health span, and so we want to have longitudinal measurements of a variety of mice so that we know what's going on. The difference here between past conventional approaches and this one is that we are not picking a preordained chronological age at which to do these analyses. And I'll talk about what the analyses are in a minute. What we're doing instead is we are doing it on a group-specific basis in relation to the % survival at the time. So each of the groups, as I said earlier, has 50 mice of each sex. And out of those 50, we wait until 10 of them are dead. And then we kill 4 of them and isolate tissues. I'll talk about that in a minute. And we also do a whole bunch of other tests on the ones that we're not killing. And then, again, for that same group, we wait until another 8 of the mice have died. And then we kill 4 of them. And then we wait until another 6 have died. And we kill 2 of them, and so on. So we believe that this is going to be a way to, if you like, increase the signal-to-noise ratio of these experiments. Because, if something's working, then you'd expect that basically health span's going to, track lifespan, more or less. So essentially you're introducing an additional variable that you already know is correlated with the ones that you actually care about. Whereas this way we're factoring that variable out, the actual amount of time that the mice live. And we therefore can see things like whether a particular intervention is predominantly affecting some aspect of function, like, liver function or whatever, rather than another intervention. We think that's useful. Hello, there we go. Yeah. All right, so this is just really written for the non-specialist on the left, just explaining the way that these kinds of things are always done, though, of course, in our case, the top box is rather more ambitious than it is in most experiments. But, there's all manner of ways to measure health span. You can do things that are visually accessible, like kyphosis and alopecia. You can do things that are behavioral, like, we can test memory. There's obviously standard ways to do that. You can do things, physiological things like rotor rod, treadmill, grip strength. You can do a whole bunch of things in the blood. So we're doing glucose tolerance tests, for example. And then there are things that you can't do unless you kill the mice. That's why we're killing some of the mice, as I mentioned a moment ago. And this is how you figure out what's going on. I want to say a bit more about that last thing, about what we do when we kill the mice. At the moment, we are not doing those experiments because, of course, when you take tissues out of a mouse, you are able to either freeze or fix them and then do your analysis at your relative leisure. And we are extremely keen to make sure that these analyses are done as usefully and informatively as possible. We're taking a lot of tissues and freezing, and some of them we're freezing, some of them we're fixing. And so, of course, we don't know everything. We are not the experts on everything. And therefore, we are very eager to go out to the community and arrange collaborations with people with particular expertise to do particular analyses. That is the process that starts today, the creation of those collaborations. I will certainly be making a lot of noise about that, not just on stage, but also on camera, on social media and everything, because we think it's very important, and the sooner we can kick off some of these collaborations, the happier I will be. So you've got my email address there if you didn't have it already. Anyone who's interested in doing such things, absolutely please get in touch. It's very exciting and important to us. I'm going to spend the last 5 minutes talking about RMR2. In other words, the next experiment that we want to do. These experiments are terrifyingly expensive, which means that we don't have any. We have only a small proportion of the money that we're going to need to do RMR2. But I'm working on that, and with a bit of luck, we'll have the requisite $3.5 million or something like that to kick such a thing off fairly soon. The important thing about these experiments is that we don't treat them as ongoing open-ended research projects, like the kind of things that I used to oversee at SENS Research Foundation. Instead, these are self-contained things. So we've paid for the whole of RMR1, or almost the whole of it, already up front, and that's what we'll be doing for the next one as well. So it makes sense for us to be thinking right now, not least because potential donors want to know, what RMR2 would consist of. And there are various options. One option is to just do different doses. We've heard yesterday, from various talks about the importance of remembering that one dose doesn't tell you all that much necessarily. Of course, we chose the doses for our various interventions that I described a moment ago on the basis of the literature, and so we're not starting completely blind, but still, we could do that. However, the question is that the most informative use of another $3.5 million? I don't think so. So that's not what we're going to do. We're also not going to wait until the mice that we're currently treating are mostly dead, and we actually have some pretty good statistically significant idea of the extent to which the interventions work. Because that's going to be a little while. We only started the experiment in February. I will say that I very much discombobulated some of my colleagues by forcing the pace of starting this experiment, but I thought it was very, very important to get it going as soon as possible, and we got away with it, as you will hear in the next talk. But the fact is I want to get the second one going soon as well. And of course, the thing we can certainly do with the second experiment is take completely new interventions for which there is also evidence of significant, tantalizing promise from the community, from the literature. Now, it won't be as robust evidence, as direct evidence as what we had in RMR1 in terms of, looking at mean and maximum lifespan, but it's, there's plenty of things that have quite a lot of evidence. So here's my list at the moment. Now, this is very much preliminary. This is the current front runners. And as you think at the bottom, it's very much front and center in my mind that the details abound. There are many, many versions on these themes, and a lot of the detail will depend on what we can afford to do. But I and my main scientist, Katie Lewis, who's here, have been spending the past maybe 3 months or more focusing on narrowing this down. I have only got a couple of minutes, so I'm only going to very briefly go through these, but you may know that a company named Retrotope has been working for a long time on a concept of replacing some of the hydrogen atoms in fatty acids with deuterium. Turns out that this is extraordinarily good for reducing oxidative stress. We want to do partial reprogramming, not just because it's fashionable, but because it's actually quite interesting. And as you heard yesterday from David Sinclair, it may be better to leave out c-Myc, but that's one thing. But again, the point is that we would do it not transgenically in the germline, but with gene therapy, same as we're doing telomerase this time round. And we are definitely looking at which of the available constructs that others have used might be appropriate. Exercise. We don't have running wheels in the cages that we're using now. It just turns out to be expensive to put running wheels in cages, and we know that exercise is quite important. We might do other types of environmental enrichment as well. We're thinking about that. You heard yesterday from Andrei Solanov about the hyaluronic acid that is expressed by naked mole rats. Of course, the experiment that you saw was done with germline genetic modification, though they made it inducible. We will want to do it with gene therapy. So we used hematopoietic stem cells, of course, for the RMR1 experiment in terms of heterochronic administration of cell therapy. But of course, many cell types seem to be quite beneficial in various ways, and this is probably largely because of the exosomes that they secrete. But anyway, so one form or another of biologically young mesenchymal stem cells seems to be quite promising. We're also pretty interested in oxytocin, has multiple modes of action, it seems, pretty good for muscle. So we've got a lot of ideas here. This is our current shortlist, and it's still pretty tentative. Love to talk about it, or maybe talk about it during the discussion. But anyway, this is what we're thinking. It's a very expensive experiment, $3.5 million at least. Oh, God, there we go. But that's it.